So let's get into translation. So far we have taken our DNA, used it as a template, one of the strands of DNA used as a template. We've gone through the transcription process, which is creating the messenger RNA. We've cleaned up that messenger RNA by removing the various introns and splicing together the various exons. And now that piece of messenger RNA, which has the 5' cap and it has a poly A tail, it's ready to leave the nucleus, head out to the cytoplasm, find some ribosomes, and make some polypeptide chains. That portion is called translation. That's concept 17.4. It says translation is the RNA-directed synthesis of a polypeptide, and we're going to take a closer look. It says genetic information flows from messenger RNA to protein through the process of translation. And this is actually supposed to be an illustration of a ribosome, which has two portions, a small subunit and a large subunit. And this kind of candy-colored strip here is the messenger RNA. And what you see here, we're going to discuss, this is another type of RNA called transfer RNA, and it's bringing in amino acids, and we're building a polypeptide chain right here. This is the process of translation. Let's take a closer look. So far we've already talked about one form of RNA called messenger RNA, or mRNA, right here. Now we're going to talk about a second form called tRNA, transfer RNA. You'll probably find that I oftentimes make the mistake of calling this translate RNA because it has to do with translation, but I'll try to recall or make sure I call it transfer RNA. But if you were to mispronounce or misstate and say translate or translation RNA, I would know what you're talking about. So this is transfer RNA. Transfer RNA, its job is to transfer amino acids to the growing polypeptide in a ribosome. So what transfer RNA does is it goes out into the various cytoplasm areas there, and it picks up the amino acids that are necessary, brings them to the ribosomes, and helps build a chain, a polypeptide chain that will eventually become a protein. Translation is a complex process, unfortunately, in terms of all the various biochemistry and parts. We're going to try to look at it fairly thoroughly, but not in every detail, so we don't want to get bogged down again in too much detail. So we have two forms of RNA so far. If messenger RNA is sort of like the recipe that you would use, well then we can consider transfer RNA like the chef's assistant who's running back and forth to the kitchen and the pantry to collect all the various ingredients. And the ribosomes maybe we could consider like the chef himself building the final product. So messenger RNA would be like the recipe. Transfer RNA would be like the helper that runs back and forth to get the various supplies from the kitchen, the ingredients. And then the ribosome itself is like the chef actually putting together those ingredients to build a final product. A great little illustration here. This brown area is a ribosome with the two parts to it. The red strip is your messenger RNA. It's being fed through the ribosome. And then these kind of bluish, greenish pieces that you see, these are the pieces of transfer RNA. They are going out into the cytoplasm, and each type of transfer RNA is particular for a specific amino acid. And here it has the amino acid attached to it. Now it is going to match up. We'll see more detail on this in a moment, but it's going to match up to its correct position on the messenger RNA, and then it's going to slide through the ribosome and attach this particular amino acid to the growing chain that we see here. And that chain is the polypeptide. It's going to become the chain that will eventually fold and form into the protein. Now you may have noticed that I said that the messenger RNAs are particular for one amino acid. So, I'm sorry, the transfer RNAs. Back that up. The transfer RNAs are particular for one amino acid. Each molecule of transfer RNA is not identical to every other molecule of transfer RNA. Each one only picks up and carries a specific amino acid. Each one has what we call an anticodon on the other end, and that anticodon is what matches up with the messenger RNA, what we call the complementary codon on the messenger RNA. So every piece of transfer RNA is not alike. On one end we have what's called the anticodon. That's kind of like the key that fits to the lock of the messenger RNA strip. And then on the opposite end we have a particular attachment point for one specific amino acid. Here we have three different models of transfer RNA. This is kind of a two-dimensional model here. This is a three-dimensional model. And this is just a simplified model that they're using in your textbook to kind of act as just a representation of transfer RNA. If you haven't done it already, I highly suggest you go watch the bioflicks on protein synthesis. These bioflicks I think are very well done, and they give you a nice overview. You can look for this bioflicks on protein synthesis in the Chapter 17 folder. I think it's right underneath there, not even in the notes portion, just under the Chapter 17 folder, along with a wide variety of links to the crash course videos and the Bozeman Science videos. All of those are very good. I especially like the Bozeman Science video for transcription and translation, and I suggest you take a look at it as well. So here's a model of transfer RNA. It's a single RNA strand. It's only about 80 nucleotides long. If we were to, it is of course 3D in shape, but we're looking at it on a 2D flat page here. If we were to flatten it out to reveal its various base pairing areas, it would kind of look like a clover leaf. So it kind of makes this shape in our model a two-dimensional model. Down at the bottom here is what we call the anticodon loop. This is where the anticodon will line up with the codon on the messenger RNA. And up here is our acceptor area that latches on to one specific amino acid. Again, here's three different models that kind of show the same thing, the transfer RNA. So you have the flattened two-dimensional model, you have kind of a 3D dimensional model here, and then you have the simplified version that they're using in your textbook. If we look at the 3D version, because of the various hydrogen bonds that occur between all of these parts of the transfer RNA, it is kind of twisted and folded in its own way, much like proteins get twisted and folded. And it kind of has a little bit of an L shape. So like if I were to spin this around, if I put this portion on the bottom, you would see it kind of has an L shape. In this case, it kind of looks like an F with just a slight little middle section for the F there. But you could call it roughly L-shaped if you're talking about the three-dimensional form. For transfer RNA to work properly, for translation to take place, you kind of have two different steps. If we go back and look at the molecule, we have two important parts of the transfer RNA. We have this section down here, the anticodon, and that has to base pair up, match up with the messenger RNA strip that's going through the ribosome. And then we have this end here called the amino acid attachment site. Both of these areas are the most important parts to making sure that translation works as it should. So first, we have to have a correct match between the transfer RNA and the particular amino acid it's supposed to pick up. And that is helped along by an enzyme called aminoacyl tRNA synthetase. I'm so sorry for these words. I would not make you memorize that, trust me. It's aminoacyl tRNA synthetase, and that is basically an enzyme that helps make sure that the correct amino acid attaches to the end of the transfer RNA. And then, of course, secondly, we have to have a correct match between the anticodon portion of the transfer RNA and the messenger RNA codon in that strip of messenger RNA flowing through the ribosome there. We do have a little bit of flexibility at one site, and that would be the third base of a codon. So the first two bases will only take the complementary base that you would expect. But in that third base, there's actually a little bit of flexibility here, and this flexibility is called wobble. This wobble actually allows some tRNAs to bind with more than one codon in that third position. We only see this flexibility in that third position, and we'll try to mention it again in just a moment. So this flexible pairing can only happen in the third codon position, and we call this ability to bond with a base that you wouldn't normally expect it to bond with. We call that wobble, and that allows for a little bit of flexibility in creating the proper polypeptide strand that we want. Of course, I think your textbook does a great job of illustration. So here's an illustration of attaching transfer RNA to the correct amino acid. So here we are, we're floating around out in the cytoplasm of the cell. Here is that particular enzyme that I told you I won't make you memorize, aminoacyl tRNA or aminoacyl tRNA synthetase. It's a particular enzyme. It has a three-dimensional shape, and it's going to take a particular amino acid and lock it into a particular initiation site. So right here, see it locks into position here, and that does take some ATP for that to happen. So it takes energy for that to happen, and it hydrolyzes a bit of ATP and releases these free phosphate molecules here. Then we have our amino acid right here. This is our particular amino acid. Here is our transfer RNA right here. So we have two different models. This is a computer model, three-dimensional, and here's a simplified model. So here's your transfer RNA, and this enzyme helps attach the two together. So here's the transfer RNA. It needs to be attached to that amino acid. This enzyme helps that process along. And then here's kind of a bit more realistic three-dimensional computer model of the process, transfer RNA, and here is that big enzyme that is helping to attach the amino acid right there. And then it gets released, and now he's ready to go. So this would be a charged transfer RNA. He is set up, he has got his amino acid, now he needs to head off and find a ribosome and match up on the anticodon with the codon on the messenger RNA. So now that our transfer RNA is loaded up with an amino acid, he is off to find a ribosome. Ribosomes are where the action takes place. Ribosomes are the actual workers that build and create these long chains of polypeptides that will go into becoming proteins. So ribosomes, they facilitate specific coupling of transfer RNA anticodons with messenger RNA codons in protein synthesis. So a ribosome is the site where the transfer RNA brings in a specific amino acid, lines up properly with the messenger RNA, and then attaches that amino acid to the ever-growing chain. The ribosome is made up of two pieces or subunits, large and small. The large one is always shown on the top and the small one on the bottom in any model. And they're made up of proteins and a third kind of RNA called ribosomal RNA, small r RNA. So far we have three different kinds of RNA, and those are really the only three that we're going to take a look at. We have the small r RNA, that's the ribosomal RNA. It's a portion, or it's part of the ribosome subunits. It's our third and final variety. We already talked about messenger RNA. That's the RNA that's created in the nucleus and then sent out to the cytoplasm. And we are still talking about transfer RNA, which is the RNA that attaches to the amino acids and brings them to the ribosomes for the protein creation process. Because most cells contain thousands of ribosomes. If you'll remember back when we were studying the cell parts, most cells have many ribosomes studded all over the endoplasmic reticulum and also floating free in the cytoplasm. rRNA is the most abundant form of cellular RNA. So this rRNA, if you were to look inside cells or a sample of cells, you're going to find more of it than you would messenger RNA or transfer RNA, just because most cells are packed with ribosomes. So there's so much of the rRNA. Bacterial and eukaryotic ribosomes are fairly similar to each other, but there are some slight and significant differences. Lucky for us, we don't have to go into detail as to what those differences are, but we might want to mention that there are some antibiotic drugs that specifically target bacterial ribosomes, and they don't harm eukaryotic ribosomes. So there are certain drugs, and you've probably heard of them before, tetracycline and streptomycin. These are antibacterial drugs that are fairly common use, that you can use to kill off bacteria because it will disrupt and affect their ribosomes, but it won't do any damage to the eukaryotic ribosomes of your own cells. So that makes it a very powerful and good antibiotic drug. So we won't go into what the differences are, just know that there are a few differences between the bacterial and eukaryotic ribosomes. We're going to be focusing on the ones that we have, the eukaryotic ribosomes. Here's a quick figure I found in your textbook. Always good to have like a nice summary. So we have our three types of RNA here. Messenger RNA carries information specifying the amino acid sequence of proteins from DNA to ribosomes. We have transfer RNA, which we're still talking about now, serves as a translator molecule in protein synthesis, translates messenger RNA codons into amino acids, carries, attaches to specific amino acids, has an anticodon tail that then attaches to the codon of the messenger RNA. And then now we've talked about ribosomal RNA, and it plays a catalytic role, an enzymatic role, and a structural role as ribosomes. So it's part of what makes up a ribosome, and it assists with the entire process of creating these proteins. So here we have a couple of different illustrations kind of showing the ribosomes. We have kind of a more 3D computer model that's supposed to be more realistic. We have the small subunit of the ribosome that's kind of in cream color here, and the large subunit in blue. We have three sites that we're going to take a look at here, and that's because we have three codons that make up the signal or the word for one particular amino acid. And we have this little strand that looks like beads would represent messenger RNA that's moving through this particular ribosome. And then we have transfer RNA molecules represented by these different colors. And then we have this chain of growing polypeptides right here. Down here we have some more simplified versions. This shows the ribosome without any of the other material. So you have the messenger RNA binding site. This is where the RNA is going to be coming through, passing through the ribosome. You have these three sites inside the ribosome that we're going to talk about. This makes up the large subunit, small subunit, and this is the exit tunnel where the growing polypeptide chain comes from. And then here we have a schematic model kind of showing the whole process. The A site, is where the transfer RNA brings in the amino acid. The P site is where it gets added to the chain. And the E site is where that transfer RNA then leaves and goes and finds another amino acid. So here's just a close-up view of each of these three models. I just like a nice big picture to kind of show you what we're looking at. So here is the probably more realistic 3D looking model. And then here's a more simplified model not showing everything, all the process. And then here's a schematic model kind of showing the process. The red strip represents the messenger RNA, and it's sliding in this case from the right through to the left. We have the the A, the P, and the E sites. The A site is where the amino acid is brought in by the transfer RNA. The P site is where the chain gets added onto. And the E site is where that transfer RNA then exits and goes off to find another amino acid to add to the chain. This little GIF or moving diagram here, if you're watching the PowerPoint, you should be able to watch this kind of move and go through the entire process. What you see right now on the screen just represents the messenger RNA and one of the subunits of the ribosomes, but it does actually move and kind of show you an animation if you're watching the PowerPoint. If you haven't seen the Bozeman Science video on transcription and translation that I posted in the Chapter 17 folder, I highly suggest you go and watch that. He does a really good job of explaining this process, and he uses this animation as well. So go check that out if you haven't done it already. As I mentioned when I was showing the illustrations, the ribosome has three areas or binding sites for the transfer RNA to come into. And if we look from the right to the left, it's PAE. So if you want to make up something that helps you remember that, it says PAE if you move from the right to the left. Wait a minute. That's not right. I'm not sure why they put these in this order. It's actually AP because the word ape kind of helps you remember moving from right to left. So let's fix that right now. Let me fix that right now. I'm going to take this out. You're going to watch me edit some PowerPoint right here. Delete that, and I'm going to place that right up here. Hit save. Alright, much better. Alright, so the original notes that you probably pulled off of the internet didn't have it in order from right to left, and now I've listed them in correct order from right to left. On the far right side is the A site that holds the transfer RNA, the one that just came in with that particular amino acid, until it can move one position to the left to be added to the chain. In the center is the P site. The P site is where the amino acid adds to the chain, the ever-lengthening chain. And then the E site is the exit site where the transfer RNA that now has given up its amino acid leaves the ribosome and goes off to find some more. So if we look right here, we have the APE. So if you want to think of it, remember you're going to be moving from right to left. Just think of the word ape, APE, and that'll kind of put you in the proper order. So this is kind of like the holding position when the transfer RNA comes in. Then it gives up its amino acid to add to this growing chain, and then it exits from the left. Just like in the process of building that messenger RNA that we're going to send out from the nucleus, we can divide the building of the polypeptide chain into three stages of translation called initiation, elongation, and termination. So we're going to use the same terminology to describe the three steps of building a polypeptide chain as we use to describe the three steps of transcribing the DNA into messenger RNA. All three of these stages require various protein factors that kind of help the process along. and keep it going in the right direction. And I did put up some notes about all of this, and I'm going to leave these notes in here, but I will give you some sound advice. Instead of reading through all of these notes or having me just kind of read through all of these notes, it's wise to look through them and look through the textbook, but even wiser still to do the following. Here is an animation about translation that describes all of those particulars of initiation, elongation, and then termination of translation. You should start right here. So take a look at this animation, watch it, and then maybe go back through and take a look at the PowerPoint slides that I have just passed over. Come back through and watch this animation again. So instead of simply going over and reading all of this information about initiation and elongation and then termination of translation, I highly suggest instead you simply watch this animation and then perhaps take a look at them again and then come back and watch the animation a second time. To me, the animation helps a lot with the understanding rather than just reading those notes. And I don't want you to bog down too much with the specific tiny little details of every particular enzyme or every particular factor that has to be in place for this process to work. So take a look at this animation, and I think it is very much better at clarifying than it would be for me just to read off of the slides. Also, again, the Bozeman Science videos, go take a look because they too are very clear with this information. So let's kind of jump ahead here. We have initiation and we have elongation, and then here it's showing termination where the ribosome comes apart. Again, take a look at that video and it'll be quite clear to you what's going on there. So now let's go to what we call polyribosomes. A single ribosome can make an average size polypeptide in less than a minute. So it's kind of a pretty fast process, but what we need oftentimes in the cell are many copies of polypeptides, many proteins. I mean, proteins are the building block of almost everything within the body there, so we need a lot of them. A number of ribosomes can translate a single mRNA simultaneously, and we call that, when they form this complex, we call that a polyribosome or a polysome. Remember I said that particularly in bacterial, in the bacteria cell, but also in the eukaryotic cell, we can have multiple RNA enzymes creating messenger RNA from the same strand of DNA, and they can kind of line up like a series of semi-trucks in a convoy. And so you can have multiple RNA enzymes creating that messenger RNA off of the same DNA strand. Well, this is very similar. You can have multiple ribosomes using the same strand of messenger RNA all at the same time, forming what we call this polyribosome, making many, many copies of the polypeptides that we need. So this allows the cell to create many, many chains of polypeptides pretty quickly, and it kind of looks like this down at the bottom. And you can actually see this polysome or polyribosome in the microscope very well. So this thin red line represents the messenger RNA, and it's sliding through, it looks like a dozen ribosomes here, and they're all creating the same polypeptide chain that you see coming out these little bluish colored dots there. I love it when you can take a nice illustration and put it right up against the actual microscope picture. So here we have a polyribosome or a polysome. It's made up of multiple ribosomes all reading off of the same messenger RNA and creating long polypeptide chains. And this one has just been finished and it's come loose, and then these are growing and being added to. And so down here we can actually see this. It looks kind of like a chain of beads, and so you see the ribosomes and you see the little strand in between, which represents the messenger RNA. This type of shot was probably done with a transmission electron microscope to get this level of detail on something so small, probably a TEM. Very nice shot here that kind of lines up perfectly with the illustration. Finally, let's talk about the end part of translation, which is forming a functional protein. Making the polypeptide chain alone is usually not enough to make a functional working protein. Some of these functional proteins are made up of multiple polypeptide chains, and even if it's made up of a single chain, it has to be modified and bent and formed into the right shape. So polypeptide chains are modified sometimes quite a bit after translation, are targeted to specific sites in the cell, taken and given to various other places in the cell to be further changed and modified until they're made into the complete protein that the cell needs. Earlier in the semester, we talked a lot about how proteins are very complex molecules. They have complex 3D shapes, so that we know after the polypeptide chain is made, even if it's a protein made of a single chain, there's going to be some coiling and folding that happens between the various bonds between the amino acids there. So it's going to take on a shape that probably doesn't look anything like a long linear chain. And then some of these proteins need to be modified by various enzymes or activated by enzymes. So there are various enzymes that will cut portions of the protein off or fold it and change its shape quite a bit. And then we know that some proteins are made up of multiple polypeptides, so there may have to be 2 or 3 chains that come together to form the functional protein that the cell needs. Some of these polypeptide chains or proteins that are being formed will be sent off to certain areas of the cell to be finished off and completed. We already know within the cell that we have ribosomes in 2 areas. We talked about the fact that there are free ribosomes, which are just floating around in the cytosol, and then we have the bound ribosomes that are stuck onto the endoplasmic reticulum. Remember, we talked about rough endoplasmic reticulum and smooth endoplasmic reticulum, and when we say rough ER, we're talking about the ribosomes that stud the ER material, the membrane there. So free ribosomes, they mostly create proteins that are going to be put to work inside the cytosol of the cell. So if it's a protein that needs to be floating around free inside the cell, it's probably going to be made by a ribosome that's floating around free inside the cell. But if it's a protein that needs to be secreted from the cell and sent off to a different cell or a different part of the body, well, it's probably going to be made by one of these bound ribosomes that's attached to the endoplasmic reticulum, because then that amino acid chain, that polypeptide chain, can be passed on to the ER, the Golgi apparatus, and it can be modified and put into a vesicle and sent out of the cell. These ribosomes, whether or not they are free or bound to the ER, they're actually identical, and they can actually swap positions. So a ribosome that's floating free in the cytosol can become attached to the ER and vice versa as well. So the way it generally works is the polypeptide chain almost always starts, or the synthesis begins there in the cytosol in one of those free-floating ribosomes, and it will finish there in the cytosol unless that polypeptide chain is designated for a particular protein that is to go to the ER and then perhaps onto the Golgi apparatus and out of the cell. So if that's the case, what will happen is that ER will attach itself, that ribosome will attach itself to the ER, and once that polypeptide chain is finished making, it will be moved into the ER and then perhaps onto the Golgi apparatus for further processing. The cell knows which of these polypeptides are supposed to go to the ER because there's a specific chemical marker called a signal peptide, and this signal peptide will bind to something called a signal recognition particle, and that SRP will bring that signal peptide and its ribosome to the ER so that that growing chain of amino acids can be given into the endoplasmic reticulum. And again, picture always helps, so let's take a look at a picture. So here we have a ribosome. Here's the messenger RNA ribbon flowing through there. Here is the chain of the polypeptide chain that's growing. On the end, there's a signal peptide that says, hey, this protein is going to be, or this amino acid chain needs to go to the ER. So this little SRP sees that signal protein, latches on, and it pulls the entire ribosome and all over to this area of the ER, and it attaches to this little spot right here called a translocation complex. And so when it attaches that ribosome, now what once was a free ribosome becomes an attached ribosome. And here you see that protein chain still growing. The little signal peptide gets broken off, cut off, because that's not actually a part of the chain that we need. That was just a signal to get it over here to where it needs to be. And then the growing chain can be made, and it goes into the ER. Here is the protein inside the ER. It has folded and taken the proper shape, and now the ER may pinch off a vesicle and send it out, or it may pass that protein on to the Golgi apparatus for further processing or something like that. So that's the basics of transcription and translation. We're going to take one more break right here. The very last portion of the chapter deals with mutations and problems. So this is obviously a very complex process. So when it's very complex, it means there's a lot of potential for error and problems. So the last part of the chapter talks about errors and mutations and problems, and we'll come back and finish up in just 1 second.